Before we move on, I want to ask Miles to please post our first poll question. So audience, please do take a second to answer, and we'll review those results in just a little bit here. But I want to make sure we also cover some more of the general information here. Daniel, what would you say are the major scientific biological advances in understanding the potential for mRNA-based immunotherapy vaccines for cancer, say, over the last 2 years? What are your thoughts on that?
Well, I think across a number of uh, elements of our business, first and foremost, I think the rapidity in which we can manufacture these vaccines. Right, what happened with COVID really showed, not only because we had the whole world behind it, but now we're starting to see personalized vaccines, like Andy mentioned, being made in a couple of months. And I don't know the exact timeline, so I don't want to make it seem long or short. But my point is this is pretty quick, and I think this technology allows for that. So on the manufacturing side, this really could be the technology we've been waiting for to help facilitate cost-effective therapeutics as we're moving forward, and we don't have to balance this, $150,000-$200,000 to make a CAR T cell. Maybe we can get that cost of goods into a realm that makes sense. So I think there's been significant opportunity there just from an operational perspective. On the science side of the equation, I think there's, again, in the cancer world, a number of things that are important. One is that there has been this underlying potential dogma that in a cancer patient, their immune response may or may not be capable of eliciting a solid anti-tumor response because the tumor has somewhat, suppressed it. And I think in the checkpoint inhibitor space, there are some patients we know that's not the case. But if you look at what, Andy and the team at Genentech and BioNTech have done in the PDAC situation, we're clearly getting patients that are getting these personalized vaccines that are generating robust T-cell responses. And so this shows to me from a scientific medical perspective that there is an immune system there that we can go in and we can really drive it. And I think the PDAC study that I read, these patients were still getting a number of other treatments like chemotherapy. So there were still suppressive elements being put into that bucket. So when you really look at the data as a whole, I think it's very exciting because there is this opportunity in these patients to really use a technology to drive it. And where we move from there, is a very interesting question.
Sure. Okay. And one of the things we do for our Cell and Gene Live events is talk to garner questions from our registrants in advance of the event. And a really great question that came in that I wanted to direct toward you, Daniel, is what are some of the major mechanism of action gaps that need to be solved in the very near future, would you say?
It's a great question. I think from my own experience, really having a better appreciation about immunogenicity around these different technologies. There was a very interesting paper in Vaccines recently. It's a Nature publication that took the BioNTech Pfizer vaccine and the Moderna one, and then essentially did an experiment where you take the different components, the UTRs, the lipids, and you essentially make hybrid vaccines to try and figure out what is the most immunogenic component to each one. And what was really quite interesting is, don't quote me exactly, but, the 3' UTR from one was better than another. The lipid from the Moderna, I think from memory, was better than the other, looking at immunogenicity. But serouridine utilization on the RNA didn't really seem to have an effect. Now, I think there is some debate around the importance of that. It's become quite entrenched, the dogma that serouridine modification to limit immunogenicity around RNA is important. But my personal opinion, and Andy probably has exposure to this, is the more you can polish your RNA and get out double-stranded RNA, and the more pure you can make your RNA, the less likely you are going to have immunogenicity. So I do think a better understanding of all of the components that make up a RNA product is needed. And, I think in the context of what we do at Myeloid, if we think about vaccines, one of the limitations there is you need to know the neoantigens. And these vaccines, I think, until we have a better understanding, are going to be limited to a neoadjuvant setting. And that is because we're still not solving for the tumor microenvironment problem, right? We're activating T cells in the periphery. These tumors are making molecules that inhibit those T cells from being activated. The T cells can't get in there. And that's really where myeloid, where we're trying to insert ourselves. By delivering CARs and getting cells into these tumors, we can change that tumor microenvironment and shift that. So I think over the next, in the near future, understanding RNA and the lipid delivery technologies better, getting away from entrenched dogmas that, may in certain circumstances hold true, but not necessarily across the full spectrum, will help. And then in the oncology setting, solving for the TME remains an important element.